Good afternoon, everyone. I hope you all had a wonderful summer and welcome back to our medical grand rounds. Thank you for all of you who are online and a large crowd in person. Welcome. For those of you who want to join us, we are in D021312 at the Glen. So before we begin, I'd like to make a brief land acknowledgement. McGill University is on land which has long served as a site of meeting exchange amongst Indigenous peoples. I acknowledge and thank the diverse Indigenous peoples whose presence marks this territory on which we now gather. So we had a cancellation a few weeks ago, and I decided that taking the chairman's prerogative that we would take over. Our research team would take over to present grand rounds today, and it's really a pleasure to come to you not only as chair of this, but as one of the presenters today. I'd like to introduce my co-speakers, Dr. Abhinav Sharma, who is an MD, PhD clinician, scientist, chercheur clinicien, and heart failure cardiologist at McGill University, whose work bridges clinical medicine and digital innovation. He leads major federally funded research programs in digital biomarkers and artificial intelligence, advancing precision medicine for patients with type 2 diabetes and heart failure. And as well, our executive manager of our program, Julie Lebel, who has a tremendous leadership experience and research experience with a background in martial arts. She applies the same discipline and dedication to her team and the participants in the research community. And finally, myself, Nadia Giannetti, a cardiologist and researcher here at the MUHC. So today we want to talk to you about the Courtois cardiovascular research program. And as you'll see, this is really not just cardiovascular. And this is a research program that has now gone beyond cardiovascular. I encourage everyone in the Department of Medicine who has clinical research interest consider working with us in this program. So this is a program which is funded by the Courtois Foundation via the McGill University Health Center Foundation, and we thank both of these foundations for their great support. And it is a program that is a research program run through the Research Institute. And what really led to us, the story behind this program was that people like myself and Abhinav Sharma and my colleagues are cardiologists that do spend some weeks rounding in our coronary care unit. And of course, when we round in a coronary care unit in an institution such as this, which is a big referral center, we see patients that really should not be there, okay? And you've seen this, you've seen headlines in the newspapers, you've all heard of that sort of 50-year-old healthy person who dropped dead golfing or running, or worst case, even running a marathon. And so about 10 to 15% of our patients in the coronary care unit I would call unexpected patients. The rest, you know who they are. They have multiple traditional risk factors, but there's this kind of smaller group which leaves us a little bit puzzled, which really, as far as we're concerned, should not be there. And that's really what led to the creation of this program. Investigators in this program are myself as program lead, Dr. Matthias Friedrich, who's scientific director, Abhinav Sharma, digital health director, and George Tanasulis, genomic director. We have an advisory board, well known to you, Dr. James J. Brophy, and others. And finally, we have a large research team, with a program manager, a lab technologist, research coordinators, administrative support, and our happiness officer on the bottom right here. So what is the objective of this research program? It is really to build a platform for collecting and maintaining biospecimens, data, and their associated information. We have blood, tissue, genomic, microbiomic, digital health, and imaging data. And the goal with this is to create a biorepository with a vision to personalize cardiovascular health. And the mission is to use cutting-edge approach to develop and validate individual signatures to optimize and personalize cardiovascular health. And this will become a little bit more clear as we go through this exactly what we are trying to do. But basically speaking, it is a personalized health program. The data that we collect in this program, many data points, cardiac imaging, cerebrovascular imaging, and including as well carotid imaging. So we image the heart, the brain, the carotids. We have a lot of labs such as biochemistry, many biomarkers, and genomic data. We have stool collection from microbiome data. We have exercise 6-minute walk test data, raw data, ECG, vitals, digital health, many questionnaires, and body measurements. So we're collecting a lot of data on our participants, and we're doing this data collection at time zero. We collect events over the years. The goal is to repeat at 5 years and then keep collecting data. We put 5 years here, but ideally in perpetuity on our participants. And using all this data and machine learning and artificial intelligence, we are identifying patterns to create cardiovascular signatures. Participant population, Dr. Sharma, who is online, I'm going to ask you to please take over for this section. Abhinav? Abhinav, I think you're frozen. Dr. Sharma? Okay. I'm going to jump in whenever you want, Abhinav, but you're frozen right now. Oh, can you go?
Yep, I'm just back. Sorry, it froze for a few minutes here. Can you still hear me? Hello?
We can hear you.
Great. Thank you so much for that very kind introduction, Dr. Giannetti. So my name is Abhinav Sharma. I'm an assistant professor in the Division of Cardiology here, and I'm a clinician scientist, as Dr. Nadia said in her kind introduction. What we need to reflect and what we really want to highlight is the strength of this biorepository. And we want to emphasize that this, while our initial focus was cardiovascular disease, this is truly a platform, if you will, for research and accelerating research across the lifespan, accelerating research across different clinical domains. And we're going to highlight some of the data points that we have and some of our recent successes in peer-reviewed public funding, leveraging this extensive database. So as you can see right here, our biorepository, which forms the overall umbrella protocol, has 4,346 participants that are available. And the specific Courtois cardiovascular signature program, which is a smaller component, or let's say a specific study of that, has 3,727 people available with a goal of reaching 4,000. And you can see the steady increase in the enrollment rates with a balance between male and females, which was very important to us overall. Next slide. What we could see is that we have a distribution of participants across Montreal Island. So as Dr. Giannetti mentioned, our data is in an open to collaborate model. So if you're interested in obtaining data, we're very welcome to discuss that with you, and I'll show you some examples of how we've done that. But to represent that, we're not just taking people from a small specific area of Montreal or those that are only in the hospital. We're actually getting a very wide distribution of individuals reflecting the demographic diversity of Montreal. So there have been researchers that have been interested in different socioeconomic variables across different regions. In terms of our populations within the Courtois Cardiovascular Signature Program, we have those who are healthy, those with risk factors, and that includes smoking, hypertension, high cholesterol, elevated BMI, and then those with established cardiovascular disease. And we also have patients with specific diseases or conditions, such as those with various malignancies, cancers, and respiratory illnesses as well. Our age is typically between 35 and 79 in order to get into the cardiovascular signature program, but there's no age restriction to get into the biorepository overall. As we can see here, there's a broad distribution of risk factors that we have with and without cardiovascular disease. And as we know, a lot of these risk factors also align with other types of disease states, such as respiratory diseases, malignancies, and cancer. And so we have a rich biorepository enriched for patients with cardiovascular disease and cardiovascular risk factors that are potentially available to researchers who may want to access this data. And here's an example of a population level analysis that we had done when looking at the distribution of hypertension by region in Canada. Sorry, here in Montreal. And we had actually initially had some discussions with the Public Health Agency of Montreal, where they were quite interested in this type of demographic distribution and demographic data. We could see that, hypertension has a different distribution across the island, as does the prevalence of elevated HbA1c as an instance. So this is one example of how our database can be used in terms of population health or epidemiologic health. But as we will show you, we can even go down to personalized medicine and personalized disease. And so let's describe some of the funding opportunities that we have been successful at. So as an example, we recently won a CIHR grant of almost $1.2 million. This was led by Dr. Judith Mandel, who was a nominated principal investigator, and I was a co-applicant to this award. And we had initially started this through an MI4 award, which was $150,000, leveraging data and biospecimens and our nurses from the Courtois cardiovascular signature program to obtain samples. The samples were then sent to Dr. Mandel's lab, where we actually did translational analysis, looking at DOCK8 dependent mechanosensitive central actin pool and how that maintains T cell shapes and protects the nucleus during migration. I'm not going to go into this in much detail, but as you could see, this small amount of funding, leveraging our database, led to a very high impact publication. This just came out in 2015, and that subsequently unlocked a large amount of CIHR funding because we had the samples on hand, we had the infrastructure, and we had the phenotyping information available for use right away. You can imagine for your own study, if you would want to access this kind of data, it would be you would have to take a lot more time to develop this. And so we can go on to the next slide. Thank you. I also wanted to highlight the Decide Omics program, and this is a multidisciplinary collaboration that has leveraged our world-class teams. If you may recall, there was a world-class teams competition of which our team came in first place amongst the emerging team, where we linked cardio, kidney, and metabolic disease together. And we leveraged the Courtois data in this program called Decide CCVS, deconstructing the phenotypic heterogeneity of type 2 diabetes associated with cardiovascular outcomes through multi-omic profiling and clinical clustering, the Decide CCVS program. This was a nominated principal investigator, along with the co-principal investigators, Dr. Thomas Mavrikanis from nephrology, Dr. Michael Tukas from endocrinology, Dr. Thanasoulis, Dr. Friedrich, and of course, Dr. Giannetti from cardio. And overall, we were able to win more than $700,000 in this CIHR competition to leverage the CCVS data in order to analyze the blood specimens we have, the blood samples that we have, to answer critical questions. And we could share that on the next slide. And so you can see how, internal teams within the research, both the Research Institute and at McGill University, spanning across the spectrum of McGill University, came together as a part of this world-class teams competition. And then we subsequently put a grant together to apply and leveraging this large data repository. And we were actually successful. So our aims for this particular study was to use data from global megatrials to identify phenotypic clusters using machine learning and see how various anti-diabetic therapies respond across these clusters. And then we would use our Decide CCVS program that Dr. Giannetti and I have been describing in detail to integrate and to enhance these clusters using multi-omic profiling. And the funds that we're obtaining will be used to run proteomic analyses, lipidomic analyses, and metabolomic analyses as well. And so that will be used to enrich the biorepository. And so these are two examples of how we've been able to leverage the database we have to develop world-class teams within the Research Institute and how we can move forward. I also want to highlight an analysis that was also approved by Dr. Judy Liu from cardiology, along with Dr. Friedrich, myself, and Dr. Louise Pilote, who were co-applicants to this award. And the title of this was Understanding Sex and Gender Differences in the Impact of Psychological Stress on Cardiovascular Disease Progression. Again, this was leveraging data from the Courtois program. And we were able to win over $500,000, approximately $500,000 in CIHR funding. So as you could see, this is an extraordinarily rich database that is now demonstrating its ability to attract peer-reviewed funding using the infrastructure we have, using the data we have, and to help both you accelerate your research and to help the community by leveraging and obtaining the samples and the data we have to launch it into peer-reviewed funding.
Thank you so much, Dr. Sharma. I know you're on CC when you have to go back to patient care. Thank you so much for that.
Thank you so much for this opportunity.
And I'd like to now ask our program manager, executive manager, Julie Lebel, to talk about the infrastructure.
Thank you. It's a pleasure to be here with you today. So I'm just going to describe a little bit of, let's say, how we built this program. So we have a space was basically shared with the foundation on the Maisonneuve, so 5100 the Maisonneuve. So we have built a beautiful clinic room, which can hold 2 participants during our visits. We have 2 ECG machine, blood pressure. We have people who can draw blood, and basically we're equipped really to perform all our visits. So we have built from scratch all our database. So one of them is Open Clinica. It is to host all our coded data. It's a validated software, so we really want it to be on the top of the clinical grade software. It's hosted in a cloud in Montreal. So we have RedCap, which holds the personal information, which is completely different, and it does not connect with the coded data. So for a secure environment within the hospital. Sorry, and the mouse has come upside down. All right, and this last one is ATSIM. It's our blood managing database. It's secured within the hospital, and we gather all the information for our blood sample in this database. So we have a beautiful MRI, which we have the MRI team here present, and it was also a donation of the Courtois Foundation. So if ever you have clinical research involving MRI, you can contact the clinical research team. And what we can offer. So the program has multiple pieces that we can offer. So first of all, we have built this amazing consent form and protocol that can hold multiple research and without having to redo everything. So we have an approved protocol that can basically be tailored to multiple research. We have database that can be also tailored to your research. We have questionnaires that the participant can complete online and also offers other researcher the possibility to create their own questionnaire within our database. We have a data sharing process as well. And we also have, with our consent form, access to medical records. So we have a really rich database. So this platform can be used to pre-screen for clinical trials. So if a researcher have a clinical trial, but instead of screening and really searching through OASIS for possible participant, they can contact us and we could pre-screen those patients. So the researcher is able to get their clinical trial and eligible patient quicker. We have possibility also to recruit other population within the other clinics. We're set up to do body measurement, ECG, blood pressure, 6-minute walk test, and as well as digital health. And also, if there's a company example who wants to test their digital device, they are able to contact us and basically test their device on our participant. We are set up as well with 2 technologists to do blood draw and blood processing, as well as biomarkers. So I'm going to talk a little later on about biomarkers, but we have a great opportunity here. We do genomic as well as fresh blood analysis, and we have the possibility to do heart MRI, brain MRI, carotid MRI, but also other body parts. So this is a machine and an instrument from Roche called Pure that we have obtained early this spring, and it allows us to perform multiple analysis at a very reduced cost. We have another platform in proteomic, which was also a donation from the Courtois Foundation. This platform is led by the proteomic group, but we also have possibility to have a reduced cost to do analysis. So I'm going to pass on to Dr. Giannetti.
Yeah. Thank you very much. So, for those of you out there who could be interested in using and having access to our data for your patients, this gives you an idea of the current patients in our biorepository. Remember, the participants have signed a consent form, and they do not need to re-sign the consent form. Okay. If somebody is interested in a specific population, for example, here, we have 465 participants with thyroid disease and 425 participants with asthma, almost over 1,000 patients with some sort of psychiatric condition. Of course, many patients with cardiac issues. Okay, a lot of patients with GI issues, whether it be Crohn's, peptic ulcer disease, irritable bowel syndrome, and on and on and on, kidney disease, cancer, osteoporosis. So if anyone has interest in having access to any of these patient populations, and for instance, compare certain patients with a control group, or compare patients, follow these patients longitudinally, or ask questions, what is the sleep pattern of patients with IBS, or what is the incidence of AFib in your patients with anxiety disorder? You can you can query the data to answer those kind of questions. Okay, so the idea is that we've created this biorepository where participants have signed a consent form allowing us to access their data. We have data samples that have been collected. We're working with researchers. We can work with companies as well if they want. And what we want researchers to do when they use the data is if they enhance the biorepository, that they put the data back into the biorepository. So the idea is to continue to enrich the biorepository. So for example, we now have added proteomics as a standard for all our participants. But let's say somebody says, I want to do metabolomics on 1,000 patients with this, and you have funding just for the metabolomics section. You would be able to access all the data, but we would want you to put the metabolomics data back into the biorepository so it can be used by other people. Okay, obviously, your data would be protected, but the idea is that you keep growing this database. Okay. There is a platform so one can access, so you can see on our website, this is from our website, it will tell you again how many participants have what. This is a live website that keeps adding the patients coming in, the risk factors of the participants. So if you go to our website, cvsignature.ca, you'll be able to access this data easily. Okay. And we really believe that, as Abhinav mentioned, that there is power in partnerships. So we really do want people to use this data, to access this data. And I would argue that if you have a small, a grant that is limited in size to study a certain patient population, you can use all the infrastructure that we have. Okay, so you don't have to start. So if tomorrow you receive $100,000 grant to study peptic ulcer disease in a certain population, rather than create the whole infrastructure, you can ask us, can I enroll my participants into your biorepository, get all the data through there, store the patient samples in your infrastructure, use the ROS machine, which gives us reduced costs. Okay, and you can use the grants that you've received really for the part that is something that only you have that we don't have. Does that make sense? Okay, so you can use the grant that you've received really specifically for the data you are collecting that we don't have in our biorepository. These are the collaborators that we are working with at the present time. You recognize, I think most of these, many of these people who are doctors that work here at the MUHC and some at McGill. And this gives you an idea of the current user protocols. So in the middle, you see here in black, the biorepository has 4,346 patients. Not all those participants are in the signature program, which is here, CCVS, which has 3,727 participants. We will be recruiting up to 4,000. We have funding up to 4,000 participants to be in the full CCVS, okay? All these other circles that you see here, okay, represent the researchers and the research programs that are already underway using the data in the biorepository. There are 35 research programs using the data. There can be an infinite amount, okay? There's no upper limit. So how does this work? Well, to access data, if you go to our website, cvsignature.ca, you can just contact us through that, okay? To access the data, you need to, send us a message through our website, and you're going to see this form. You can fill out this form that says the kind of population you want to study. The first thing we'll do is we'll tell you, excuse me, how many participants we have with what, the condition that you want to study. And then from there, you need to write a user protocol. And in the user protocol, it's a 2-pager, which includes, background, rationale, objectives, design, duration, sample size, thank you, and the population that you want to study. And the user protocol goes to the ethics committee. So remember, the participant doesn't have to re-sign. But it's not, it's not open access. It's not a big buffet that's sort of open for anything. Every study protocol does have to go to ethics. And ethics judges, the merit of the study, whether it's ethical, to look at this data. All of this data is then provided to you in an anonymized way, obviously, and then you can redo your research. But it does require an ethics approval. It's usually quite short. It's not a lengthy ethics approval. It's usually about a 2 months ethics approval. And then from there, we can give you the data to perform your study. Now, I think what we have built is just the beginning. And together we can transform this foundational work into discoveries that personalize cardiovascular health and deliver real impact for patients. And today I want to show you where we are at. Remember, this is called a signature program. Mitchell, are you online? Yeah.
Yes, I am. Hi,
Yeah,
Nadia.
Mitchell, I don't know if so we are working with, Mitchell is a PhD engineer who has developed a company called Area 19 that we have been working with, and Area 19 is officially a company. We have given them a small amount of data to begin. And with this small amount of data, we are able to now see the signature of our participants. The first early, we call this signature 1, okay? Now, Mitchell, can you go live here?
Yeah, if you just stop sharing your screen.
Stop
I can
sharing.
actually-
Okay, so I'm going to ask Mitchell to show us.
Okay. I hope you can see the Cardiome dashboard.
So just so you know, every single participant right now, we have signature 1 on, okay? We have not given it to participants yet. This is now in ethics, so we're waiting for the go-ahead to be able to release these signatures to participants. So Mitchell, go ahead.
All right, so what you're seeing here is an interactive dashboard that will be accessible to all of the participants and also any researcher that's interested in looking at the signatures.
Mitch, can you talk louder? Mitch, talk a bit louder, please.
Yeah, certainly. So what you see here is a cardiovascular wellness score that's based out of an extremely sophisticated, heterogeneous, and explainable AI system that we developed at Area 19. So each participant gets access to a dashboard like this, where you see the overall cardiovascular wellness score, but we also break it down into 4 subcategories. So diet quality, lifestyle, physical health, and mental well-being. And the actual signature is this panel here. So it's pretty easy to interpret. Basically, everything that's above 0 is good. It's working in favor of this person's cardiovascular wellness. And everything that's negative is working against it. So very quickly, you see for this participant, the major factor is exercise frequency. And as it happens, this participant is only working out or moving 2 times a week, which can certainly be worked on. And what's really interesting is that in real time, this participant can adjust what ifs. So, what if I increase my exercise frequency from 2 to 5 times a week? In real time. The signature is recomputed, and then we recompute the cardiovascular wellness scores for this. So very easily, you can do different scenarios where you can work on specific points that need to be worked on. So this is very interactive, and we also provide these cardio populations. So this positions each participant with respect to the rest of the population. So for example, and for each of the biomarkers. So this is LDL cholesterol. This person's is in the healthy range, but it's a bit close to the unhealthy threshold. And that's one thing that the cardiome signature picked up on. LDL could be worked on a little bit here, and so on and so forth for diet and lifestyle. But what's really cool is that we have an AI system that can prescribe the optimal recipe for that person. This will show what has the most impacts, what areas can be worked on that have the most impact to improve the score to a maximum of 86% for this person. So exercise frequency was recommended. The optimized target is 6 times a week. Total cholesterol, some modifications there, slight increase in the exercise intensity, and then modulating the LDL to bring that down a little bit. And it goes into a very narrow detail, very fine-grained details. It even recommends for this person to increase the grain consumption, which will have a marginal impact on the wellness scores. So if I reset this, frankly, this has been extremely interesting to play around with. You can see we can see what the impact of changing the amount of potatoes a week has on your wellness scores, oily fish, plain white fish, nuts and seeds, et cetera, et cetera. So this gives you a really fine level of detail into controlling things that you can control that can have, cumulatively a very big impact on the cardiovascular wellness. So like I said, we're waiting for ethics approval to release this to all of the participants, and we're looking forward to working with the PIs and with the participants to optimize this and hopefully help them optimize their health.
So much, Mitchell. So this is something that we're referring to as signature 1, which is because we've just started, we don't have the long-term follow-up on our participants. Our first participant is hitting year 5 like imminently. So we're about to start the 5-year testing, and this first signature doesn't have some of the data points yet that we have collected. So this is signature 1. It gives you an idea of what you can do. Area 19 already had pre-existing data, and this is what is added from the Courtois signature program to form this signature. So I'm going to stop there. I thank you very much for your attention, and I hope that many more of you will work with us to use this very rich data set in the future, and we can all advance personalization of healthcare together. So thank you very much for your attention, and I'm open for questions. Are there any questions in the room? Yes, Fed.
Great talk and great initiative.
Do you want to maybe, I'd like people to hear, so I'll just, Ben Smith has a question if you want to just grab a microphone.
Great talk and work by all of you. Lots of potential. As part of the recruitment or selection process, are you including some version of controls or people without disease that brought them to hospital?
So we do have, so we do have a third of the population is considered healthy. Okay. So you do have a nice control group to work with. So we do have some people who, some of them may have other, but certainly not cardiovascular, and they would fall into our healthy population. So the answer is yes. Okay. Any other questions in the room? Yes. Just one.
The last set of data you presented with Signature One program. So is it also intended for the patient to try to get more involved in his health and try to improve his health? If so, his health status might change along time. So how will you take that into account in your studies?
Yes, that's something that's imperfect. You're absolutely right. First of all, the residual risk is also not account. When you change a parameter, you have your residual risk from, let's say, your cholesterol has been high your whole life, right? So there are limitations with this kind of signature. So the residual risk and the change over time. So that is one of the issues, okay, that we've dealt with, because right now this is not meant to be an interventional study, okay, which is one of the reasons why it's sitting in ethics, okay? It's not an interventional study. And perhaps one of the things that we will do is give people their signatures, but not give them, the ability to make changes. In other words, not as detailed as what Mitchell showed you there. Okay? Yes, that's definitely an issue, and that's a limitation of this. If halfway through you do change people's behavior, their exercise, their diet, adjust their cholesterol, so that's a problem. There's no way around that, okay? So the timing has to be very clear when you release these signatures, and that's one of the reasons that the signatures have not been released. The idea of doing an intervention, the idea of changing your data set, okay? Something that we are can't really hard to mitigate, because once it's out, okay? So just I didn't mention, but we do. The other thing that we do, which does affect this, the data, is that we do give back critical values. Okay, so if we find someone has an ejection fraction of 20% on MRI, we will let them know. So there are very extreme, what we call critical. So critical labs, if your potassium falls at, comes back at 7, we're not, because we see this data. In fact, patients can see it because it's on the dashboard. Okay? So that makes the biorepository imperfect in that the extremes, the criticals are dealt with. Okay, and that's one of the limitations of a biorepository, of a long-term biorepository like this. Yes, Thomas, yeah.
Nadia, great talk. Just a question. So you said the CV signature stops at 4,000 participants.
That's the funding we have for up to 4,000, yeah.
But the biorepository, there is
endless.
Endless, okay. Yeah, that was the clarification. Thank you.
So what Dr. Mavrokanis is asking is the actual, all those data points that I showed you that we're collecting, the MRIs and the stool samples and everything. We have funding for 4,000 participants. At 5 years, we repeat all the testing. So we have funding out to 10 years. If someone tomorrow wants to come and say, I'm going to be doing CT scans on, patients with asthma, can I put that into your the biorepository is open. Okay, And one of the challenges is to, make sure that we can keep funding this into perpetuity. We haven't talked about that, but for now it's funded through granting and philanthropic. But at some point it has to be funded more by grants, less by philanthropy. Okay. So we want this biorepository. There's no limit, upper limit of the biorepository. I'm going to go to the questions in the chat. I have something by Sasha Bernatsky. If we need a cardiac MRI, cost per patient, volume timeline. Cardiac MRI is something that for an observational study, I'll let you speak directly to Julie about that. That is one of our limitations, is access to cardiac MRI. So if you say, could you expand your biorepository to 6,000 participants, it would be very difficult because we're going to start MRI 2 on our first participant, and it's been calculated such that at some point, if we were to do more cardiac MRIs, we'd have to do them outside because the MRI is pretty much saturated. Sasha, you can speak to Julie Lebel at muhc.mcgill.ca for details about cost. Dr. Rosenblatt, I think Dr. Tobin, you had your hand up. Patricia, Dr. Tonin, Patricia, you had your hand up. Did you want to make a comment verbally?
Sure. Yeah, I think it was a fantastic presentation. It's enviable. I've been trying to do this with OncoDrive for quite some time through the breast clinic. My question is the genetic data. If you could comment on what type of genetic data you have, first of all, whether it was derived from targeted sequencing, whole genome, whole exome sequencing of the germline. The second thing is, if I wanted to have access to DNA, do you offer DNA so that if you don't have this data in your repository, but we are doing this genetic sequencing and then we will put it back into your repository, is that possible?
Totally. So to answer your first question, it's something we're working with Vincent Muser at the Genome Center. George Tanasulis, who's well known to you, is our PI for genetics. It is called Exome Plus. Okay, it is not full, whole genome. It's it's more of a 200 than a 2,000 per participant cost. But it is quite extensive, but it's not whole genome sequencing. The answer to your question is yes. If you wrote a user now you would have to have funding for that, right? You'd have to say, I have funding just for the DNA. Can I access that? So the answer is yes, through a user protocol. I don't make the decision. The ethics committee says, yes, this is valid research. And yes, so the answer is you can access the blood. Right, as long as you're within the RI, we're not sharing blood with companies, but you as a researcher at the RI can access the blood for the testing that you want, as long as ethics has approved your user protocol.
Okay, thank you.
Welcome. Dr. Rosenblatt, how many data sets like this? Okay, so look, in terms of Cardiologist, first of all, Matthias Friedrich, who is one of our PI, has worked, and some of our PIs have worked with Cardiologist. There is no data set that is as extensive as the signature program. There are many biorepositories, UK Biobank, lots of biorepositories out there. This is not the biggest by far. It's considered a small biorepository, but it is deep. It's a really deep dive. So there are no other biorepositories that exist that have this kind of detailed, if you look at, your proteomics, your microbiome, all the MRI, and Dr. Friedrich does a very special MRI. He could give you an hour presentation just on the oxygen sensitivity MRI. The MRIs are done without injection, but there's a certain kind of MRI that's done, which allows looking at allows you to look at microvascular. So it is unique in that deep phenotyping, certainly not in numbers. Okay. Okay, so the website, thanks George Perlman, who's one of our researchers, has given you cbsignature.ca is the website. Dr. Wiseman. Yes, so to answer your question, as I mentioned that, Jeff, so the only incidentally discovered anomalies, and we have discovered some, we've discovered, the Novoy fib, we've discovered patients with blood pressures of 200 systolic, we've discovered patients with cholesterols of 10. We have very, 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 very well-established criteria to deal with what's called critical findings, okay? We call the patients and we say, we have found tumor in your head, okay? We have called patients to say, or on the spot, your blood pressure is 200. So anyone that has a predefined critical, and it is predefined in our protocol, they are informed, they are sent to a doctor, it is dealt with. Things that are kind of more in the middle, it will show up. The patients can go to the website, they have a personal link, and they can get their data so they can find their laboratory work as an example, okay? And then it becomes up to them. We're not here to be their physicians, but we do inform people of critical findings, and that is one of the limitations of the biorepository. Dr. Pilote, factions based on semi-site incorporating. Louise, did you want to unmute yourself? Yeah.
Yes, thank you.
Yeah.
Yeah, I was very impressed with Mitchell's presentation of the signature, which this was all about. So it's nice to see. If you increase your activity level by so much, you will, improve your, scores and all that. So that requires, estimates to evaluate the impact of that change. And the estimates, I just wonder what they're based on. There's so many trials that have obtained those estimates. So it's I'm wondering how you obtain the estimates and what they're based on.
Mitchell, are you able to answer that question? Mitchell, are you still there? Okay, all I can tell you, Louise, is that all I can tell you was this a lot of this was pre-populated with data from clinical trials, external data, and then when we came in with our data, so unfortunately I would need Mitchell to tell you exactly what data has gone into the algorithm, because this cardiome is an algorithm. It's been 5 years in the making. Okay, that's a very good comment. Unfortunately, I don't have the answer offhand, but I'm happy to have Mitchell reach back out to you. Can the data structure and algorithms be shared with either biorepositories? Congratulations. Yes, Ruth. Yes, absolutely. We can work with other biorepositories, and we are in touch with other biorepositories. So absolutely. Dr. Doss. Don't think we have this data, Dr. Doss. I would be happy to speak to you offline. I don't have anything about plastic nanoparticles and mass spectroscopy, so that's not part of this. Are you enrolling? Okay, Dr. Khan, are you enrolling First Nations or Inuit? Absolutely. Who owns the data? The data is all stored at the RI anonymously. There is no, in terms of ownership of the data, it's really sitting at the RI and owned by the RI. But we do have First Nations and Inuit participants. There are a minority. We're talking about maybe 3 to 4% that have enrolled in the program, and their data is treated the same as everyone else's data. Yes, Ben. Yes, come to the microphone. Yeah.
The events, ascertainments that happen between these visits, is it only events that occur within the MUHC, or you can also ascertain events?
That's a good question. We send questionnaires. So every year there are questionnaires, and we harass participants. Okay. And we harass participants, and so the answer is we rely a lot on questionnaires, but we do have somebody who is really running through the data, okay, because obviously not all these patients are seen at the MUHC. So there are questionnaires every year, any new medical conditions, any new medications, anything new, it's there. We have had to drop some of the original participants who were not responding. So when we're looking at the 4,000, these are really, and that's why you see that one of the reasons the biorepository has more than 4,000, because we still have some data on some of these participants. They've been taken out of the actual signature program, because the signature, we need participants who are following up, and we need their data over time. Okay, so we rely on the annual questionnaires. We will really go for, we really go after patients to make them. We have a emergency contact on everyone in the case of death because we want to know what the cause of. We already have some deceased participants. We already have participants who've developed cancer. We've already had participants who've had heart attacks and other endpoints, right? So I didn't show you this, but we do have some endpoint data that is coming in, okay? And we have somebody who combs through the clinical data to pull out. Okay. Any other questions? Okay, well, I hope some of you will join us in this journey of trying to create personalized health signature for patients. Oh, Sasha's hand is up. Last word to Sasha.
Ariane has a question too.
Ariane, sorry, I didn't see you. Ariane, you're there? Ariane, go ahead.
Thank you, Nadia. This is really amazing. It's a real tour de force to see this all coming together and to see this incredible infrastructure. You know, the challenge is obviously finding sort of the unifying science strategy that will enable the sustainability of the platform and really sort of choosing the lens that you're going to bring to how you investigate all these different disease models. So I think that's going to be really fun to see how this evolves into the next phase. I had a specific question. You started out, and I remember when this was going on, there was a whole thing around sudden cardiac death and unexpected death. In your sample, do you actually have, what's the number of patients that you have who have sudden cardiac death and have survived, or are you actually doing a risk profile?
Yeah, absolutely. I don't remember the number offhand, but we have, and that's the number I'm always looking for, Ariane, because yes, you and I have discussed this many times. So we do have our group of unexpected patients. It's in the hundreds, not more than that, okay? But we do have our group of those really catastrophic type patients. So we do have sudden death, and we do have unexpected participants, which I'm pushing very hard to always make sure that we go to the CCU and we get those people who are there for no apparent reason. So the numbers are there. It's the minority. It's in the hundreds, certainly not in the thousands. Be happy to review that with you, as well as the actual sudden death number. I can get all that information to you.
Okay, thank you.
Thank you, Sasha.
Sorry, Dr. Khan has a better question than mine, asking about enrolling First Nations or Inuit and ownership and so on.
Yeah, it's all owned it's really all at the MUHC. So we're coming up on the hour. Any other final questions? Otherwise,
so I will ask my question then. So as I understand it, you've kind of got controls, like relatively healthy people. You've got people with heart disease and people without heart disease, but it's enriched in people with heart disease.
So a third have heart disease. Okay. A third are what we call healthy, and a third have some risk factors. And by risk factors, I mean more the traditional kind of risk factors.
I just wondered, because you mentioned I think you mentioned things like if you wanted a sample of people with irritable bowel syndrome as an example, or it could be anything. So the question I wondered is like the sampling frame, is it reflective of the general population of people with anything you want to think of? Or do you have to kind of think about
people, believe it or not, I think, I didn't emphasize this, but we have not had to work really hard to recruit participants. Okay. This is, we've publicized quite a bit. We have those little cards out there. People are enrolling because a lot of it is word of mouth. Okay. So those 4,000 participants, they're coming to us. So there's a lot of people just coming from, as Abhinav showed, kind of all over. Word of mouth, patients, family members, a lot of, people who work in the hospitals, a lot of people who work in the hospital, people who are on, attending these rounds who are participants. So it's kind of a mix of people. We have, made sure to maintain that about 1/3 has cardiovascular disease, 1/3 is healthy, and the other group sits in between. So if you wanted a specific population, let's say you want, your 500 patients with bowel issues, I can't tell you where they fall. We'd pull them out, and then we'd be able to tell you what comorbidities they have.
Right. I just wondered in terms of if you said I just want your irritable bowel patients, I guess as researchers, we have to consider whether they may be may or may not be more likely to have certain conditions than in the general population. But this is great, and I love your transparency and your enthusiasm for involving other people. Thank you so much.
Thank you, Sasha. So thanks everyone for participating, and we'll close out the session. Have a great day and see you next week. Bye-bye.